Welcome friends once again to my module of NPTEL on exploring health data, healthcare survey data. We are on the verge of completion since we are dealing with the last week of the session. I hope you must have been enjoying with our all hands-on experiences on healthcare data. This week we already initiated estimation with the use of evaluation techniques. I know that this is the need of the hour where we are evaluating various programs. The government and even many NGOs, many corporate houses are targeting you to engage the researchers who have repository of knowledge with evaluation techniques. That is how we have thought of giving a module where you guys have enough understanding of evaluation techniques. Yes, given the paucity of time, we are not giving the detailed information about all those techniques, but we are for sure giving you the capsule requirement that may address important dimensions of evaluation designs and techniques. We are here on the last week, and 2 lectures we have already taken on on evaluation models or evaluation techniques. First one was on introduction, second one was on PSM. Here we are discussing about randomized control trial. We know that no introduction is required to explain randomized control trial. However, it is good to mention and refer the latest Nobel Prize that was given in 2019 to Professor Abhijit Banerjee Doklo and their team. And so you must be surprised to know that nowadays around 80% of the research in developing and undeveloped countries are utilizing RCT designs or evaluation techniques. So, since they are using this, obviously the person who is having knowledge on this will be most demanded. So, let us go further. Understanding of randomized control trial. RCT is trial in which subjects are randomly assigned to one of the 2 groups, that is called experimental group and another is called control group. But in PSM we are not dividing it by experimental group or control group. Though we are saying there are some counterfactuals and treatment group, but here we are specifically emphasizing on these 2. Okay, that is why the PSM was defined as the quasi experimental, but this one is in fact called experimental design. So, what do you mean by experimental group? Experimental group is one receiving the intervention that is being tested. What do you mean by comparison group? They are also called control group, which are not receiving the intervention treatment. So, RCT can be used to measure both program intervention, that is nutritional supplements distributed as a measure of nutritional program, and policy interventions. Okay, the like cash distributed as a result of cash transfer policy, etc. Okay, so largely this is used to identify the intervention of certain programs, maybe on nutritional supplements, how this has impacted to the larger population. We can understand the implications of policy designs or policy interventions. So, this is defined as like what is our sample and how exactly the sampling frame is defined. There are 2 groups is defined from the population. One is the treatment group, another is called control group, and in both the case we will have certain outcomes, and those outcomes has to be compared. Subjects in a population are randomly allocated to groups, usually called treatment and control groups. The results are assessed by comparing the outcome in the 2 or more groups. To ensure that the groups are being compared are equivalent, respondents are allocated to them randomly. That is what we have been saying several times. The numbers allocated to both the groups should be randomly allocated. There should not be any single biasness. So, that means those are present in each of the groups by chance. Okay. Within the limits of chance, randomization ensures that control and treatment groups will be comparable at the start of an investigation. RCTs can often provide the strongest causal evidence about a program's effectiveness. So, RCT tests the extent to which specific planned impacts are being achieved. One of its strengths is that it provides a very strong or significant response to questions of causality, causality and helping evaluators and program implementers. So, this provides the cause and effect relationship and also helps the evaluator as well as the program whoever are trying to implement. And this helps us to know that what is being achieved is as a result of the intervention and not anything else. RCTs first took hold as a research strategy in the medical field. They largely apply RCTs design. Today, they are widely used for a broader array of social sciences and social policy design as well. The design of RCT is as follows, starting with the population, then there are selection criteria, one is called participants and another is called non-participants. So, like participants who meet the criteria and those are non-participants who are not at all part of the program, like if cash transfer program is being evaluated, we are considering the population who are eligible for cash transfers. Who are not at all eligible, they are not part of our group for RCT design. Then within the eligible group, we have to define as treatment and or the experimental group and non-experimental group. So, then those who meet the criteria, basically those who meet the criteria of cash transfer program, they are considered. Then invitation to participate, some may refuse, they are also subtracted. And their consent should be taken, thus it was important, this has to be followed. So, once they are they are denying, then there has to be crossed, they are not relevant for us. Then we go to actual participants who are actually relevant for us. Then we will divide into control groups and treatment groups. Then that is possible through the randomization technique. So, when randomization, perfect randomization is understood, that means we are actually following the design of RCT correctly. So, now come to the time of allocation in the randomized trial study design. First is a subset of the source population is identified to be potential members of the study population. They could be source population, could be study population and remaining population. Second, a sample of potential participants are assessed for inclusion criteria. So, like a pre-testing or a sample of potential participants should be taken and study participants are also selected. So, accordingly study participant and non-participants could be understood. From the sample further we can subtract who are not actually the participants. Then investigators randomize study participants and assign the treatment. Now, we can compare treatment group and then we can have a comparison group. At predetermined intervals during study follow-up, investigators assesses the outcome status for all participants. Then further, like for treatment group cases and non-cases, then comparison group cases and non-cases, then further stratification can also be made, further design can also be made to compare the results. The simplest RCT design has one treatment group and a control group, that is what, as simple as possibly defined. Variation on the design are to have either there will be multiple treatment arms or there will be factorial design, there may be sequential factoring or maybe a clear arms are defined, maybe a structure is defined instead of a clear chain of the factorings. In case of multiple treatment arms, for example, one treatment group receive intervention A and second treatment group receive intervention B, accordingly we can classify. In case of factorial design, in which a third treatment arm receives both intervention and intervention B. Like individually we say intervention A and intervention B in each of the case, and maybe a third design, factorial design, where both there might be some cases where both intervention could have been taken. Coming to the experimental design further, it is basically a type of experiment that has a treatment group and a control group. In case of experimental design, we need to compare those with the receiving group, basically the treatment one, and to their non-receiving group. And we have to also make sure that the cases observations are randomly assigned, okay, in both the group, and we need to take relevant measurements of both the groups when treatment is complete. Then what do you mean by randomized assignment? That is what is the focus of the RCT tool, RCT design. So randomization is quite important. So random assignment equitably ensures that every eligible units has same chance of receiving the program, and free of subjective criteria or any sort of reservations has to be avoided. And it is randomized assignment given that a sufficient large number of units to which the randomized assignment process is applied. Randomized assignment will produce groups with statistically equivalent averages of all their characteristics. Okay. These averages should also tend towards population averages, okay, ensuring that the study is representative of the broader population. So there might be some issues here, like if your average treatment, average estimation we do, if that is not experimented to different groups or different population, larger population, your result might not be applicable to a larger context. So we have to make sure that as well. So what about the control variables in this case? This includes the variables that can reduce the variance and increase the precision of the impact estimates. When outcome variables are correlated with observable factors such as age or educational level, in that case control variables are actually important. This helps in reducing the variance and increase the precision of the impact estimates. The most important control variable to include in the analysis of RCT data is the baseline level of final outcome variable. So, the baseline level of the final outcome variable is most important to emphasize. However, including covariates that are influenced by the treatment can treat bias in the estimates. Okay. In addition, including too many control variables is likely to reduce rather than increase the precision of the estimate. So, too many, too less has to be taken care of. This RCT design usually means that control variables must be collected before randomization occurs. So, before randomization you did, those control variables must have been thoroughly observed. Before the program begins, researcher can use baseline data on the population of interest to verify that there are no systematic differences in observed characteristics between the treatment and control units. Therefore, any differences in outcome between the treatment and control groups after the program can be explained only by the program itself. Now, coming to certain aspects like called intent to treat ITT versus treatment on treat, that is called TOT. So, we are explaining called ITT and TOT. When designing RCT, the researchers must decide whether to estimate the ITT or TOT. So, that is on treatment on the treat and intent to treat. So, ITT and TOT or both. The ITT estimates the average effect of offering the treatment on outcomes. This is basically the average effects regarding the treatment. The TOT treatment on the treat, those are the treatment group. The treatment on the treat is estimated. TOT estimates the average effect of the actual treatment on the outcomes. or the effect only on those who receive the full treatment. So, those who receive the likewise in PSM we discuss about ATET as compared to ATE. Here also there is a difference between ITT and TOT. Okay, TOT is specifically on the group where the actual or full treatment is taken or given. In some cases where program participation is voluntary, the ITT may be the more policy relevant effect. Okay. Like if program participation is quite voluntary instead of it is specifically targeted. When it is voluntary, that means there are huge randomness attached. So, it is better to evaluate both the groups. Okay. When it is involuntary or very specific, then you have to differentiate who are the specific persons willing to take the program and how what really happened to them. Okay. So, researchers may be interested in understanding the effect of the intervention on everyone in the population. So, in that case the ITT is maybe ITT may be considered. What about blinding in RCTs? Blinding is in RCTs is discussed as like randomization does not eliminate the confounders that may sweep in after the allocation has taken place. So, the confounding variables, the confounders may have also impacted. They might create some blinding on the results. So, blinding can play a major role in controlling these post randomization confounders. Okay. So, after the even if you have done certain randomization with the assumption that there is no difference between these two groups, but still due to the confounders, the control variables that might have actually impacted. So, blinding plays an important role. Blinding is a measure in randomized control trials. So, blinding is a measure in RCTs to reduce detection and performance bias. Okay. The differences in care or attention provided to the subjects in different arms are referred to as performance bias. If the researcher has foreknowledge about the treatment assignment of a study participant, it may influence his or her judgment in outcome assessment, which will lead to some detection bias. So, if there are some prior understanding, okay, that may lead to certain detection bias. Those could be also identified through the RCT design. Blinding refers to the act of masking on the nature of treatment that participants in a RCT receive. A single blinded trial, it involves blinding of any one group of individuals. Usually the subjects receiving the intervention or the outcome assessors are blinded to the intervention assignment. What do you mean by then? That is basically called single blinded trial, and as compared to the double blinded trial. The single blinded one where this involves blinding of any one group of individuals. Okay, whereas in case of a double blinded trial where any two groups of individuals are actually blinded. In case of single, usually the subjects receiving the intervention or the outcome assessors are blinded to the intervention assessment. So, these two differences has to be noted very carefully. But in case of double blinded one, it is recommended in usually in drugs trial. Wherever drugs trials are taken, both have been actually blinded and they do not have any sort of information about the kind of intervention is going to be given. Usually the investigators as well as the study participants are blinded to the allocation status. Who has been allocated, nobody knows. Okay. There are triple blinded trials as well. Okay. The participants, the investigators and the person analyzing the data are all blinded. It is not about the participants, it is investigator also. So, what is actually being run or whoever are analyzing these contexts, they also do not know. So, finally, there might have been some codes assigned, assigned to the person. So, which code carries for what purposes nobody knows, but we can accordingly get the result and then comparison is made. So, this must be very complicated for sure. So, then now you might have understood how randomization is done. So, then why to randomize? It is quite obvious that we will understand the non-biased component, okay, in the experimentation. So, randomization allows researchers to identify a group of program participants and a group of non-participants that are statistically equivalent in the absence of a program. In ensuring statistical equivalence between groups, randomization rules out confounding variables. Okay. So, this will rules out confounding variables, and also statistical equivalent results without or with the absence of a program can also be compared. Simple before after comparisons or comparisons between non randomized groups are likely biased by factors outside the program itself. That is why randomization is needed. So, all these 3 points regarding randomization is equally important to note. RCTs overcome these issues to provide a reliable estimation of program impact. If the two groups are identical in all aspects other than their participation in the program, then any differences in outcome must be accredited to the program itself. If they are completely identical in all aspects and they are also randomized equally, then any kind of outcome that is derived obviously would be resulted due to the program itself. Okay, that is why randomization is needed. Random assignment versus random sampling. What do you mean by that? So, it is a obvious question that that should be verified. So, is it the random sampling that is called random assignment? We need to clarify. Random assignment should not be confused with random sampling. Okay, random sampling refers to how a sample is drawn from one or more populations, whereas random assignment refers to how an individual or a group are assigned to either a treatment group or to a control group. So, basically it is not just the persons to be randomly picked up, rather we are actually dividing into two groups. Okay. How they have been categorized, if that is follows with random assignment without having any bias, that is basically called random assignment. RCTs typically use both random sampling, since they are usually aiming to make inferences about larger population, and random assignment. So, both random sampling and random assignment because of an essential characteristics of RCT. So, RCT as well as random sampling both are used in RCT design because of making or result more populated or more representative. So, what do you mean by what is the level of randomization? Randomization is typically done at the level at which the program is implemented. So, for example, assignment to community health program will be randomized at the community level. If it is at the household level or at the district level, then accordingly district level randomization is made. Note that randomization cannot take place at a very lower or disaggregated level, okay, because there will be lots of possible biasness. Okay. So, usually all kind of program evaluation technique is taken by the JPAL group. Okay, JPAL group in their maximum program evaluation techniques, they usually do it at the district level. Okay. So, if it is lower than that of the community level, then it might not find the randomization appropriately. Okay. Then individual versus clusters, how randomization is made. In an individual randomization, individual units are assigned to a treatment or control group. In cluster randomization, clusters of units rather than the units themselves are randomly assigned to treatment or group. So, basically we know that clusters like cohort study are made. So, cohort once we are defined, the cluster itself are selected instead of any person within the cluster are selected to both the groups. Cluster may be a village. Okay. Likewise, in NSS in census data, cluster villages were defined. Okay. So, accordingly the randomization are done. Clustered RCTs are the preferred type of RCTs when the intervention is by definition applied to the cluster level rather than at the individual level. that is an intervention targeted towards health facilities in a given setting. If it is a cluster based, a given setting is given, how health intervention actually implicated at a community level or at a cluster level. The statistical power in cluster RCTs is typically lower than that of the individual RCT. Okay. So, that has to be noted down. Since outcome within clusters are typically somewhat similar to each other, so the differences cannot be actually identified across the clusters, but across the individual the cluster impact of RCTs or any program can be identified. Cluster RCTs are often more expensive than individually randomized RCTs. These are more expensive as well. However, cluster RCTs provide administrative convenience, okay, and reduce ethical concerns and avoid treatment group contamination. What do you mean by phase in randomization? In phase in randomization, the rollout of the intervention is randomized and every unit or cluster in the population of interest will get the program eventually. For example, in an intervention intended to treat around 100 villages, 50 villages are randomly selected to receive interventions in year first and 50 villages selected to receive intervention in second year, okay, and then comparison can be made. The latter group that is in the serve as the control group in year 1. Okay. Phase in designs also reduce concerns of inequity and provide incentives to maintain contact. However, for control participants, phase in designs could change present actions through setting expectations of future change. Phase in designs complicate estimating long run effects, since once the intervention is fully rolled out, no control groups remains. Okay. So, regarding data collection, RCTs require baseline as well as endline data and may also include midline data. Some in between data is required to have a proper flow of information for better designing in the second one or for better implementation of the RCTs. These data sets can span the course of weeks, months or years. This data is often collected via primary data collection method. Okay. So, there are steps involved in conducting RCTs. Steps like one is like need assessment, program theory, baseline survey, randomization techniques, okay, monitoring, some follow ups, okay. So, etc. are involved. In case of need assessment, and this is an optional prelude. An optional prelude is a need assessment which can provide information on the context and its constraints. That is, as an example, need assessment could tell us how many children have received their full immunization course in rural Rajasthan, if focus is Rajasthan, how many have received. So that means if we wanted to know about the number of children, then need based assessment is made. A program theory, this is also known as logic model, causal model, intervention logic is actually developed. A program theory explains how an intervention is understood to contribute to a chain of results that produce the intended or actual impacts. A baseline survey is conducted of the entire target population or entire target sample. Data are collected on the relevant indicators. A sample should be randomized into different groups, as we already mentioned. Randomization can be done using the softwares like Excel or Strata to ensure, or randomized table as well, to ensure that randomization has succeeded and check they are and check that they are equivalent in terms of baseline indicators and contextual variable that might be important in the study. The program or intervention is implemented in the treatment group. During the program, it is strongly advisable to monitor the program's implementation. Okay, so monitoring is always essential in program implementation design or program evaluation design. Following the program's implementation and depending on the context of the evaluation, end line and follow up survey should be conducted. Some indicators are quick to respond and others may slow. So, intervention is going to give you right direction. Outcomes are compared between treatment and control groups to determine the impact estimate. Results are reported to the implementing partner once this process have been completed. Okay. So, then couple of things to be guided to you to understand this very correctly. We are just coming to the explanation of thread to design how to read at the design correctly. First one is called spillover and crossover post thread to design. There are some spillover and crossover issues. Spillover occurs when a program changes outcome for units in the control group. Okay. If that program gets changed and have some outcomes in the control group, then the spillover impact may create some problem. Spillovers may be physical, behavioral, informational, market or general equilibrium based, whereas a crossover occur when a control unit directly receives the program either intentionally or accidentally. By any reason, if there are some crossover, then it is very difficult to go by the RCTs. In both cases, the comparison groups no longer serve as a counterfactual. Okay, so, they are no longer a counterfactual where the treatment cannot be actually evaluated. So, when is it appropriate to use ICT? This is very interesting to be noted. RCT should be planned from the beginning of the program. Entire structure of the RCT is very essential while going for RCT based evaluation. RCTs need a large sample. Okay. This is another important aspect. This can only be used when the sample size is big enough to detect effects of the program with sufficient precision. The study design must have what statisticians call sufficient power. Power is the probability of correctly concluding that an effective program is in fact working. Then again we need to understand what you mean by power calculation. This is basically to conduct and calculate the required sample size for an evaluation. Okay. Evaluators must use assumptions regarding the expected effect size and the statistical significance level and the intra-cluster correlations if you have any sort of cluster RCT design. Intra-cluster correlations is basically a descriptive statistic between 0 and 1 that indicates how strongly your groups, those are household based or if it is individual in the clusters, resemble each other in both the groups of comparison. The higher the intra-cluster correlation, the higher is the required sample size to be considered for RCT design. RCT should be undertaken following formative research or evaluation. RCT must be appropriate to the nature of program being assessed. RCTs are best used for programs that seek to achieve clear measurable impacts that can be attributed to a distinct intervention and which lend themselves to certain causal pathway for better analysis in case of program implementations. Some couple of issues at the last are very essential, like understanding cost of RCTs, how the expenses are borne, that has to be decided because you are evaluating comparing groups and the programs whether received by them or not, and there are so many interventions as well, like behavioral issues as well that may have certain crossovers. Okay. So, cost is quite essential. RCTs are known to be the prohibitively expensive method, since they have come to be synonymous with hard evidence. Numerous government and NGOs have invested in them and donors have been willing to fund them because they give concretized information. This incur cost because of staff cost, data collection method, intervention cost, overhead cost, utilities, time lag as well. Okay. The complexity and scale of the RCTs would determine how complex these buckets are in hand and of themselves, along with factors such as sample size, the design as well as the duration of the study. Nonetheless, we should have ethical consideration, because we are directly observing the persons or the clusters, So, their ethical considerations should have been taken. There should not be having any sort of conflicting or competing interest. Okay. Their identities are at stake. So, some ethical considerations should be taken beforehand. RCT has been criticized on the ground that randomistas, they are also often referred to as referred to as this word as randomistas, are willing to basically sacrifice the well-being of participants in order to learn. Okay. It is often pointed out that due to randomization, people who need a certain treatment do not receive it, okay, while others have a treatment and they do not need. So, that is very interesting. So, some who do not need, and where in some case they have got it, but they did not have need. In some case they received it, okay, they did not receive it even. Okay. If households from a village are selected randomly for an intervention while others remain in control group, it could lead to disruption in community, because some may assume that why they are selected, why and I am not selected. There might be possibility of further conflicts and violation within the community. Okay. So, after explaining all sort of arrays of or chain of discussion in RCT, we have discussed about its design, its steps, its randomization technique, its I.I.T and TOT, and its cost context, then its ethical consideration at the end is always discussed. With this, I am quite sure that you might have received huge understanding related to RCTs. Further details on it, we are going to add it in our next episode, not on this call. Okay. It will be discussed in our next year maybe. This time we have clarified the basic design of RCT, their advantages, their disadvantages, their limitations. The practical side of applying to a data, it requires a direct field observation and it involves cost. Okay. Therefore, we are also limited in explaining all those aspects. So, if you have any queries, do not hesitate, and we will try our best to address to you through our team. They are Mr. Milind and Mr. Kamal. They will be also proactively helping in this regard to get a direction in this design for program evaluation or evaluation based methods. With this, I think it is time to stop, and we will explain further details in the next class. Thank you.